So let's dive in a little bit to how proteins fold and are stabilized. So as a disclaimer, it's not super well understood how protein folding happens. We know it kind of starts locally, we get those bits of secondary structure starting to form, and then the secondary structures come together to form the tertiary structure. In this process, water is excluded, and then eventually these proteins find their way to the lowest energy conformation, which is kind of what I want to show you with this image here on the right hand side. You see that these proteins kind of start completely unfolded, then maybe some parts of secondary structure start to form, get us into a lower energy structure, and eventually we find that lowest energy structure, this protein's native or normal structure. Now I'm going to show you a video that kind of in real time shows how a protein is folding. So in this video, what you'll see is that the first thing that happens as we find a lower energy conformation is that some aspects of secondary structure start to form. See here, we have a little bit of an alpha helix forming at the very bottom, and maybe that's a beta sheet up on the top here. Now, as we get closer and closer to the lowest energy state of this protein, you see more aspects of secondary structure has formed, and then additionally, we have a tertiary structure where these secondary structures have come together to make the final fold. And so what do we know about what stabilizes protein folding? This is largely controlled by non-covalent interactions. In particular, the important one, folks, for protein folding in tertiary structure is the hydrophobic effect and van der Waals interactions, London dispersion forces. Ionic interactions and hydrogen bonding, of course, do play a role, but the most important is the hydrophobic effect. Why is this true? So we should imagine this protein surrounded and dissolved in water. So here's our protein, and all around it we have water, right? We would therefore expect, since this protein is surrounded by water, that all the interactions with water are favorable. So we'd expect polar amino acids that can do dipole-dipole or hydrogen bonding with that water, or charged amino acids that can do some ion-dipole interactions with the water, maybe forming some nice hydration shells. All of the internal residues that are hidden away from water we would expect to be non-polar. So think of these as forming kind of like the core of this protein. Why does it make sense that they should be non-polar on the inside? Well, as this folding is occurring, all the nonpolar bits go to the center and minimize their interactions with water. This follows the hydrophobic effect because then we would expect the entropy of water to be increased as much as possible, since we no longer have unfavorable interactions between nonpolar R groups and the polar water. So this is what's really motivating and stabilizing this tertiary structure and fold. Now, of course, there are ionic interactions that can help stabilize structure, and there are hydrogen bonding interactions that can stabilize the structure, but they are less important. When we're talking about or anticipating which parts of a protein will be on the inside or on the outside, we can use something called a hydropathy plot to help us. A hydropathy plot basically plots the residue number or the primary structure against polarity. So higher up on the plot, hydrophobic residues, lower on the plot, hydrophilic residues. So since we know the hydrophobic effect controls protein folding, we would expect most of these residues to be on the inside or the core of the protein, and we would expect many of these residues then to be on the outside and interacting with water. To wrap up protein folding, let's talk a little bit about misfolding. In this class, we'll be discussing structure and function, right? The structure of a protein often tells us a lot about its function. Well, if the structure is incorrect, if it's misfolded, the function is no longer going to work either. And this is the root of many protein-based diseases, such as Alzheimer's and Parkinson's, that we believe are caused by the misfolding of protein.